Welcome back, life science learners. Let's continue with our lesson. A recap of where we are. We're focusing on nutrition today, and in that process we're looking specifically at the process of ingestion and digestion. We've discussed already the physical process of digestion. Let's now focus on chemical digestion. Remember that chemical digestion involves the process of enzymes and other chemicals that assist in breaking down the food. The objective of chemical digestion is to be able to break down complex food molecules into the simplest molecules. This is then going to be absorbed in the next process. So after chemical digestion, we have absorption. It's important that we recognize that absorption takes place of the simplest monomers or building blocks of our organic molecules. So the best way to break them down is to use enzymes and chemicals to reduce them to their monomers, and then these are absorbed. So having said that, let's go into the process of chemical digestion and unpack what are the enzymes, what are the substrates they work on, and how they are broken up into the simplest monomers. Right, so as I mentioned, chemical digestion is the breakdown of large food compounds into smaller food molecules by digestive enzymes. We spoke about that firstly being broken down by mechanical digestion into smaller bits. Now we're moving from these smaller bits into breaking them up further into even smaller bits, and that's where we want to get to. We want to get to looking at how these are then absorbed as their simple monomers into the bloodstream. So most food particles are too large to be absorbed from the alimentary canal, and hence it needs to be chemically digested, and that's essentially the important part that we're looking at. To assist all of this, we have enzymes. Enzymes are very sensitive to changes in temperature, pH, and only work in an optimal temperature and pH range. Having said that, we've got to understand that throughout the digestive system and the alimentary canal, from the mouth down through the esophagus into the stomach, we have different pHs. What does this mean? It means that the enzymes that function in these different organs or parts have to be very sensitive or adapted to specific pHs. So in the mouth, we've got enzymes that break down your carbohydrates, and the pH in the mouth is slightly alkaline. However, when we move to the stomach, the pH changes radically. As we mentioned early on, the pH in the stomach is highly acidic, and so that enzyme nature has to be an enzyme that functions at an acidic pH. It's important that we recognize how the body has structurally suited itself or adapted itself for this process of chemical digestion in the alimentary canal, where there are significant changes in the pH along the digestive system. So let's look at the table below that provides a summary of the action of the enzymes. As I mentioned earlier on, it's important that you make notes of these. This is an important section in chemical digestion, where we look at the enzymes, we look at what are the substances that they target, and what happens when they break these down in terms of the products. Right, so the table below shows the different enzymes, where they are produced. Again, it's important to understand where they are produced, the substrates in terms of what they target and they break down, and then the optimal pH and the end products. So it's quite a comprehensive table, and the best way to learn these enzymes or to have an overview of the different enzymes is to be able to refer to such a table. Again, it talks about the enzyme, the substrate, its preference of pH, and then finally the monomers that these complex molecules are broken down into. So as I mentioned, we're going to look at these groups of enzymes. We're going to look at the carbohydrates, the enzymes that break down proteins, and the enzymes that break down lipids. So these are our major groups of organic compounds: carbohydrates, proteins, and your lipids. Okay, so when we talk of your carbohydrates, they are produced firstly in the mouth. So we have saliva that contains your carbohydrates. Salivary amylase is a group of carbohydrate breaking down enzymes. We also have them produced by the pancreas, as well as in the intestine. So the pancreas releases pancreatic juices into the duodenum, and we also know that the intestine releases enzymes that help in the digestion of your carbohydrates. Okay. The substrate that these enzymes work on would be carbohydrates. So all of these saliva, pancreatic juices and your intestinal juices contain enzymes that break down your carbohydrates and your complex starch. The preferred pH for all of these enzymes is slightly alkaline, so above 7, so this could be between 7 to 9, and that's slightly alkaline where the chemical digestion of your carbohydrates take place. And finally, all of the carbohydrates are broken down into the simplest monomers called glucose, and it is then that the glucose can be absorbed into the bloodstream. Okay, so if we were to look at that again, the enzymes produced that break down carbohydrates are in the saliva, in the pancreas, as well as from the small intestine. Again, they target your complex carbohydrates such as starch. It also breaks down your disaccharides in terms of into the simplest monomers, which we know is glucose. The next group of organic compounds we're looking at are called your proteins. And proteins are broken down by a group of enzymes called proteases. Again, remember that the suffix ases often refers to enzymes that assist in breaking down. So your proteases target your proteins. So we have the first process of protein digestion starts in the stomach, where the churning of the food with the gastric juices produces chyme. We also see that the pancreas produces some important protein digesting enzymes, along with the intestine producing enzymes that are responsible for protein digestion. The substrate that all of these target would be proteins, and so we find that in the stomach, the large protein molecules, so your beans, your meat, your fish, your steak, are mixed with the digestive juices and then broken down into smaller polypeptides. Okay? These then move into the stomach and the small intestine, where the pH changes radically. As I mentioned, the pH in the stomach is acidic, so the gastric juices producing hydrochloric acid allow for those protein digesting enzymes to function optimally. However, when the chyme moves into the small intestine, we see a change in pH now that becomes alkaline. So it's between 7 to 9. It means that there's a radical change from being acidic in the stomach to being alkaline in the duodenum. So to be able to assist this, we have the pancreatic juices that produce the alkaline enzymes to adjust the temperature, and that moves from now the stomach into the small intestine. And finally, your proteases, as I mentioned, protein digesting enzymes, breaks down proteins into the simplest molecules called amino acids. It is these amino acids that then travel along the digestive system, absorbed into the bloodstream, and then move into cells where they're used for protein synthesis in a process called assimilation, or putting them together. And finally, the last group of organic compounds are your lipids, and these enzymes that break down lipids are called lipases. So lipid digesting enzymes are called lipases. The lipases are produced by the pancreatic juices in the pancreas, along with the intestinal juices produced in the intestine. These enzymes target the lipids, and remember that lipids are made up of fatty acids and glycerol, so these break them down into smaller molecules of fatty acids and glycerol. These enzymes all function in an alkaline medium. So from the mouth into the stomach, we saw that it was very acidic. From the duodenum further through the alimentary canal, the small intestine and the large intestine, we see that the pH now is back to alkaline, which is slightly above 7 to about 9. And finally, the building blocks that fats are broken up into would be your fatty acid and your glycerol molecules. And it is these molecules that are then absorbed in the small intestine and transported to where the body needs them, and they're absorbed. Right. So this is essentially what we've just looked at as a larger image. Let's look at the chemical digestion of proteins, carbohydrates, and lipids as an overview. So as I mentioned, your protein molecules are complex molecules made up of different types of amino acids. During the protein digesting process, the proteases break down the peptide bonds into a series of steps, releasing the simplest monomers called amino acids. And so that completes the protein digestion, where amino acids are released. A starch molecule, again, is a complex carbohydrate made up of many glucose molecules. Your carbohydrates break down the glucose molecules, releasing each monomer, and these glucose molecules are then available for absorption in the bloodstream. As we mentioned earlier on, a fat molecule is made up of fatty acid and glycerol molecules. Again, lipases break down the ester bonds, causing them to be broken up into the glycerol and fatty acid molecules, which then move into the bloodstream to be absorbed and used where needed. Let's look at the overview of this. So as I mentioned, the esophagus, in the mouth you find salivary amylase being produced, and that then travels down the esophagus. In the stomach, we've got your proteases, which break down proteins into polypeptides. Polypeptides are smaller molecules that make up proteins. We know that in the stomach, chemical digestion takes place, and this mixture of food along with that is called chyme. And so those digestive juices from the stomach with food is called chyme. The chyme then moves into the small intestine, which is the duodenum, along with enzymes produced by the pancreatic duct, then mix with the chyme to neutralize that pH. So the pancreas then produces amylase, lipase, protein digesting enzymes, your nucleic acid digesting enzymes called nucleases, as well as your bicarbonate ions, and those are enzymes and ions that neutralize the pH from acidic chyme into an alkaline secretion. We also see that then the food is then broken up further in the small intestine as we look at that further. Let's not forget the function of the liver. The liver produces bile salts, which are temporarily stored in the gallbladder, which are then released via the bile duct into the duodenum, that helps in fat digestion. So guys, as we wrap this segment up, it's important that we have an overview of the application of some of these concepts. So I've put a few questions together as we wrap this segment up. Let's see if we can apply what we've just looked at. So the question refers to the following image that I've got here up on the board. Study the diagram of the digestive system and answer the questions that follow. So we've got a diagram that has various different parts labeled on it. Provide labels for parts A to K and 11 marks. Guys, this sort of question is a very good question to be able to revise this section. I'm going to give us a chance to do this together and to help you to recap some of those basic parts that we did. As I mentioned, use these diagrams to recollect structure, and then also, along with that, annotate the function. So let's help me do this together. So we've got this tube that brings food down, and that tube there, A, is the esophagus. And the esophagus, again, is that tube that allows for the ball of food to move into organ B, which is the stomach. So note, as I'm going through the digestive system, I'm making note of the different parts. The food then mixes with enzymes and then moves into part C, which is the duodenum. So it's the first part of the small intestine. This leaf-shaped organ here, which plays an important role in the production of enzymes and hormones, is called the pancreas. And that's the pancreas here, which produces hormones as well as enzymes. And finally, we've got structure. Here, structure E, which is the small intestine, and that's the middle bit of the small intestine. So you can call that the small intestine, which then moves food into the large intestine. Here, we see that as the ascending colon, the transverse colon, and then E, will be the descending colon, with finally food being temporarily stored in the rectum, where it is released through an organ called the anus. Further up here, the distinct shape of this triangular organ points to the liver, and this little small gland there is the gallbladder. Guys, that's a wrap for this segment. We've looked at the parts of the digestive system. We've looked at two important processes, ingestion as well as digestion. When we looked at digestion, we broke that up into the mechanical digestion, where food was mixed and chewed and churned. And then finally, we looked at chemical digestion of the three major organic groups in terms of how proteins are digested, how carbohydrates are digested, along with how lipids are digested. Guys, that's a wrap for me. Trust that you've enjoyed the lesson and that you're able to engage with this topic as you plan for your exams. From me, have a good day. See you soon. Cheers.